Your lesson today is on using a Punnett square. The learning objective is that you can predict the outcome of offspring between two parents using a Punnett square. First, some vocabulary review from last week. You don't have to write this down, it's already in the notes you took last week. Remember that alleles is the word we use to represent forms of genes that are in a person, and you have two alleles for every trait. One came from mom, one came from dad. And we call that your genotype. It's represented by letters of the alphabet. And again, each letter of the two, one set came from mom, one came from dad. And then that determines your physical appearance, also called phenotype. And that's what gives you the trait, either the dominant or recessive trait. So for instance, blue or brown eyes. More vocabulary: to be homozygous or purebred means you have two of the same alleles, two dominant or two recessive. And to be heterozygous or hybrid means that you have two different alleles, one of each. So we use a Punnett square to help predict Mendelian genetics, basically inheritance patterns that follow the rules that Mendel came up with. Some inheritance is more complex than follows Mendel's rules, and so you may or may not use a Punnett square for that. And Mendel himself never used Punnett squares, but his results that he came up with would have been following the same rules that, the same probabilities that you see with the Punnett square. So again, just a little bit of background about Punnett squares. You don't have to write this down. It's named after Reginald Punnett, the man who created them, and he just used it as a tool to help organize what inheritance patterns we see between parents. So he actually reproduced quite a lot of Mendel's work with peas. He was a professor at Cambridge University in England. He wrote a book called Mendelism. And basically this Punnett square showed up, he created it in the early 1900s. And it just helps us organize our thinking about what's happening between two parents. So also, before you start to use a Punnett square, you have to create this 4-squared box if one's not given to you. And again, the most important thing is to know what's going on with the parents' genotypes. And if the language is hard for you, then that will be the hardest part of doing a Punnett square. So here are your steps. This is the first thing you're going to write. You have to figure out the parents' genotypes. So there's two parents. One is going to go across the top, and one will go down the side. It does not matter which parent you put where. They are interchangeable and you get the same results. But figuring out those parent genotypes is, again, probably the most tricky part. You just have to know your vocabulary pretty well. And just one letter goes in each box of the two alleles. So the next step would be to basically fill in the squares by dropping letters across and down the boxes. So if we imagine that one parent is big A, and the other parent is little a, then the only combination that can happen is big A, little a. So all of their babies would be hybrid organisms. So let's say the parents are both heterozygous for brown eyes. We're going to use letter B, looking at eye color this time. So let's imagine or think about what's the genotype you would assign. Hopefully you came up with big B, little b, one of each. And what would be their phenotype? Well, if you know eye color that's dominant, then this would be brown. So we're going to fill in our parents, big B, little b, big B, little b. And then we're going to carry the letters down and across into each box. So in this first box, two big B's. Next to that, a big B and a little b. Down here in this box, big B, little b. And in our final box, two little b's. So now you're going to analyze the results. So this is where the probability piece comes in. And we know that in this cross, or in any cross of Punnett squares, there's 4 possibilities, right? So the results are always out of 4. So if we're going to look, how many of our babies are going to have 2 brown alleles? So 1/4, this box right here, we'll just shade this in, are going to be 2 big B's or brown eyes. 2 out of the 4 boxes, these two, will be big B, little b, which are going to also have brown eyes. And 1/4, this one down here, will be blue eyes, 2 little b's.
Dear Tim and Moby, my parents both have brown eyes, but mine are blue. What's going on? From Tyra. Well, I bet there are blue-eyed people somewhere in your family tree. Eye color is just one of many physical traits we inherit from our parents. That passing of traits from parents to offspring is known as heredity. But remember that parents inherit traits from their parents, and so on, as far back as your family tree can go. Let's look at some pea plants to see how it all works. Yes, pea plants. Every living thing inherits traits. Back in the 1800s, a monk named Gregor Mendel used pea plants to study how traits like height, color, and shape are inherited. When Mendel crossed short plants with tall plants, he noticed that only tall plants were produced. Whatever produced these short plants seemed to disappear. Mendel called that tall height trait the dominant factor because it dominated or covered up the short height form. He called the short height trait the recessive factor. By the second generation of pea plants, those recessive short plant forms cropped up again. After tons of research, Mendel was able to predict that, on average, 1 out of every 4 pea plants would be short. Each plant has 2 genes that determine height. The tall gene, represented here by a capital T, is dominant, which means it only takes 1 of these genes to make a plant tall. It takes 2 short genes, represented by a lowercase t, to make a plant short. In the 19th century, a British geneticist named Reginald Punnett also experimented with peas and developed something called the Punnett Square. Among other things, this Punnett Square lets you predict the likelihood of what you would get if you crossed a tall plant with a short plant. An offspring gets 1 gene from each parent, either a tall or a short gene. Each plant produced by these 2 plants will be tall because each one has one of those dominant tall genes. But what happens if 2 of these new plants produce offspring? There are lots of ways this could turn out, but the most likely result is that we'd get 3 tall plants and 1 short one. Mendel only figured this stuff out after carefully studying 30,000 individual pea plants over an 8-year period. Well, I guess it's pretty reasonable to assume that he had peas for dinner every night. I think you're missing the point. Moby? Moby?